Hello students, myself Shashikala Maitri, working as Assistant Professor at Gokaraju Rangaraju College of Pharmacy, Hyderabad. Today we will learn about plant tissue culture. So from this topic, we will be able to define what is plant tissue culture, will understand about the term totipotency, and you will be able to explain callus culture, and you will be able to explain the various steps involved in callus culture. Let us see what is plant tissue culture. So plant tissue culture is in vitro cultivation of plant cell or tissue under aseptic and controlled environmental conditions on semi-solid or into liquid nutrient medium. The main application of plant tissue culture is production of secondary metabolites or to regenerate a plant. The success of this plant tissue culture relies on totipotency. Now, we'll have a question. What is totipotency? How you will explain this term? Just pause yourself and write the answers for this question. So once you complete your answer, then play the video. So what is totipotency? So totipotency is nothing but, as you know, it is an inherent capacity of plant cell or tissue to develop into an entire plant. Is it? It is a capacity of the plant cell to develop into an entire plant. So this totipotency you can explain in two terms. One is dedifferentiation and another one is redifferentiation. So what is dedifferentiation and redifferentiation? So here the explant which is taken from the matured plant, or it may be any part of the plant, it may be leaf, it may be stem, or it may be any part of the plant. So when it is taken, it is inoculated on the medium. When it is inoculated on the medium, by utilizing the nutrients present in the media, it undergo dedifferentiation. That is, the mature cell utilizing the nutrients from the media, it will undergo meristematic state, and when it achieves the meristematic state, it will form the callus, which is nothing but dedifferentiation. So here you cannot make any part of the plant, it is just present as a mass of the cell. And when it is subcultured on new medium, it undergo redifferentiation, that is, it can grow in the form of the plant. So these two, dedifferentiation and redifferentiation, are inherent in the capacity of the plant cells, which will allow any part of the plant to grow in the form of the plant. So now, what are the types of plant tissue culture? If you classify this type of plant tissue culture, depending upon the medium, there are two types, callus culture and suspension culture. There is one more classification which is based on the parts of the plant used. So different parts of the plants are stem, leaf, root, flower, etc. So based on those, there are different types of classification. So here we will see there are two types mainly, callus culture and suspension culture, based on the media. So let us see what is callus culture. So callus is undifferentiated mass of cell. You can see in the picture. So it is an undifferentiated mass of cell. So it shows many growing points. It can be produced by inoculating any part of the plant, stem, leaf, root, which are known as explants. So these explants are inoculated aseptically on solid or into liquid medium. So what is the nature of this callus? So the callus may be soft or it may be hard, that is, it may be pressile or compact. It varies in coloration, it may be cream colored, white color, or it may be purple colored. So this coloration depends on many factors such as light, type of media used, hormones which are provided in media, and the type of the culture conditions. So now we will see what are the steps involved in callus culture. The various steps which are involved in callus culture are mainly there are three steps: selection and preparation of culture media, preparation and inoculation of explant on selected media, and incubation of cultures. So let us see these steps one by one. Preparation of culture media. So how the media is prepared? So here 4 stock solutions are prepared. Stock solution 1, which consists of magnesium sulfate, potassium bisulfate, potassium nitrate, ammonium nitrate, calcium chloride. Stock solution 2, which consists of boric acid, manganese sulfate, zinc sulfate, copper sulfate, cobalt sulfate. And stock solution 3 consists of ferrous sulfate, sodium EDTA, and stock 4 consists of inositol, thiamine, then pyridamine, nicotinic acid, and glycine. So all these 4 stock solutions are prepared separately. Then to prepare the soluble media, these stock solutions are taken in appropriate ml. So here nowadays synthetic culture media are used. The synthetic culture media, here you can see in this chart, like White's media, Murasik and Skog media, Gamborg media, N6 media, and Nichis media. So these are the ready-made media which are used nowadays depending upon the requirement. So commonly used culture media is Murasik and Skog media. So now the next step is preparation and inoculation of explant. So how the explant is prepared? Let us see what is explant first. So explant is nothing but it is any part of the plant. As I told in the previous slide, it may be root, it may be stem, it may be leaf, or it may be meristematic tissue like cambium, or it may be floral parts like anthers, stamens, it. So these are act as an explant for the production of callus culture. So how to prepare the explant? So by using these different tools, scissors, scalpel, knife. So using these different tools, the explant about 1 centimeter or 1.5 centimeter are prepared. So once the explant is prepared, they are sterilized with different chemicals like chromic acid, mercury chloride, calcium hypochlorite, sodium hypochlorite, and then alcohol. The treatment with these chemical agents should be very less. It may be around 1 minute or 2 minutes. So once the explants are sterilized, then they are transferred aseptically onto the defined media. So next step, the last step involves the incubation of these cultures. So once the inoculation is done in the culture media, the cultures are transferred to the BOD incubator where the conditions, the physical conditions are maintained. The temperatures like 25 plus or minus 2 degrees Celsius and about around 16 hours photoperiod and 8 hours dark is maintained, which is very necessary for callus production. So callus produced within 3 to 8 days. So these are the steps which are involved in the production of callus. So you can see in this figure the steps involved in the callus culture. So here the plant is taken. From the plant, the explants, the plant is selected. From this, the parts are taken which are surface sterilized. Then they are transferred to the medium. There is a production of callus and then it is transferred to the medium for the further growth. It may be for the callus or it may be for further reproduce in the form of the plant. Here they transfer to the another media which will give the microshoots. Then it will grow in the form of the plant which can be potted in the pot. So these are the different steps which are involved in the callus culture. So in the summary, from this topic, now you will be able to tell what is plant tissue culture, what is totipotency, what is callus culture, and what are the steps involved in the callus culture. Thank you.